In this video we will discuss about some of the sources of CAR-NKT cells. So far in the previous video when we are discussing the papers previously or one of the previous paper, our focus was mainly on the T cells. That is how we can optimize the CAR T cells so that it can give the optimum functions. And in the end we have discussed that we can increase the efficiencies of these CAR T therapies by combining it with other immune cells. In this video we will discuss about the NK cells, that is natural killer cells, and how these natural killer cells can also be modified and formed to have a CAR-NKT cells, and this can also perform similar to what we have seen in the T cells performance or CAR T cells performance. So in this video we will discuss about the different sources of these natural killer cells to make these CAR-NKT cells. So one of the common or main sources, this peripheral blood. So one can directly take the natural killer cells from the patients and isolate them and engineer them in the feeder cells. That will help in the expansion of these NK cells, and then do the modifications to make the CAR-NKT cells, and that can use for the treatment against the tumors. The other ways is to use this induced pluripotent stem cells, and we have discussed this approach for the T cells, but this approach can also be used for the these natural killer cells, where the feeder-free differentiation will be used to make iPSCs, derived natural killer cells, and they can be used similarly that they can be used for the similar modifications that we have did in the CAR T cells to make them efficient to work against the tumor cells. And then one of the most common ways and easiest way to use these natural killer cells is to use the NK92 cell lines. And this NK92 cell lines, one of the advantage is that we can modify these NK92 cell lines and store them in the liquid nitrogens or in the lab in advance. However, before using these NK92 cells directly into the patients, we need to irradiate these cell lines so that it does not cause another secondary malignancies within the patients. And some of the changes, or one of the major difference between NK cells and the T cells is that NK cells have the natural ability to secrete lytic granules, whereas T cells does not have the natural ability to secrete lactic granules. And through several studies it has been known that one of the common ways to make sure this CAR-NKT cells has enhanced proliferations and persistence in the in vivo conditions is to induce this IL-15 gene into the NK cells, or knock in this IL-15 gene in the NK cells, and this IL-15 genes will help in the expansions of these NK cells or CAR-NKT cells. At the same time, it will also activate this CAR-NK T cells to release the lytic granules such as granzyme and perforin against the tumors. Along with that, the other receptors can also be modified. For example, this CD16 that has the informations about the antigens of the tumor cells, and this CAR that also contains the targeted tumor antigen informations, and it can be customized based on the patients or the tumors which are found in the patient. So these are some of the common receptors that are shown here. However, this CAR-NKT cells is not limited to just to these receptors, but other receptors can be modified as long as they will serve the purpose of increasing anti-tumor efficacies against the tumors. So here the advantage, NKT cells, one of the advantage against the T cells is that the cells can directly be used from the NKT to cell lines, whereas in the case of T cells, right now we have that kind of cell lines available that can directly be used in the patients. So this is one of the advantage of this, using the natural killer T cells. And another advantage is that it has the natural ability to release lytic granules, whereas T cells does not has that ability. So I hope you understood the sources for the CAR-NKT cells and the differences between its difference from the CAR-T cells. So if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.